Hey, Maria, or Mariah, sorry. Good morning.
Good morning.
Good morning. Good to have you.
Huh?
Good to have you in class.
Thank you. Nice to meet you.
Yeah. Right. Okay. There's chat.
You sound kind of staticky.
Um, yeah, maybe a little bit. But you can hear me, right?
Yeah.
Okay. Right. Okay, we want to go to downloads. Well, that. Okay. And we're going to want the syllabus. Let's get that. T8. Sure. Yeah. Hi, Benita. Morning. I'm doing good. Glad to have you here.
Good morning, Mr. Converse.
Good to hear from you. All right. Okay, let's get here. Okay. Yabus, good to have you here.
I'm sorry, were you talking to me?
I was.
Oh, I'm sorry. What did you say?
I was just saying, glad to have you in class. Glad to
Oh,
see you.
thank you.
Mhm. All right, we got 3 more people. There's Mariah. Okay. Two more and then we're ready to go.
All right, I was able to get in from my phone.
That sound better?
Yeah.
Oh, okay. All right. Just got two more people and then we can start. See where they are. Yeah, people.
I'm gonna go and grab my coffee.
That's fine. That's fine. Amy. Mhm.
Forgot to even turn on my coffee pot.
Oh, that cold coffee won't go down well.
Yeah.
Let's see. No, for the calendar. Go. Go. How to, send it. All right. They've been messaged. Very good. All right, y'all. Let's begin by beginning. So this is microbiology, and the objective of this course is to sort of introduce you to organisms that you can't see with your naked eye. Okay. And you might think, well, what sort of organisms are those? Well, bacteria, fungi, like mold, and little creatures that can cause disease that are called protists. Okay. So we're going to explore how each of those are built, what each of those do, and then we're going to distinguish between the good ones and the bad ones, right? The good ones help us out, or they don't have any effect on us, and the bad ones make us sick. Okay. So that's the idea, is to understand, since you're going into a clinical role, about the organisms that make you sick, how they do that, right? And how we fix it when they do make you sick. Okay. And that's going to involve knowing how the organisms are put together and knowing where their weak spots are. Okay. And also understanding how they make you sick, right? How do they get in? What do they go after? What do they hurt? Okay. And why does that make you ill? Okay. So that's our course title and our prereqs. Okay. We're here for 10 weeks. We're going all Zoom. That includes lab. But in lab, what we're going to have is I'm going to send you activities to complete and send back to me. And that'll be the component of your lab grade. Your lecture grade is going to be a combination of some presentations you're going to do, but also mostly connect work. Okay. And I'll show you where all that's located. Now, I also offer weekly office hours, Friday from 5 to 7. And there's a separate Zoom link for that, and I'll send that to you. And that's to ask me about anything that you have a question about, whether it's this class or any other science or math related class. So you just come in, you tell me what school and what subject, and we go to it. Okay. So I'm good with medical math. I'm good with anatomy and physiology. Obviously, this class, I'm good with pathophysiology, which is the changes in the body that happen when you get sick. Okay. And I can help you with basic arithmetic and algebra. All right. So whatever help you need, you come to me and we'll work on it. Okay. That's my time for you. And that's every Friday, 5 to 7 p.m. Okay. If you want a separate time for sitting down and tutoring, working with me, let me know and we'll arrange that. The course objectives are here, right? We'll go through them one by one, right? Talk about the adaptation, attenuation, structure, and resistance of bacteria, fungi, parasites, and viruses. What does that mean? Well, the structure is just how they're built, what they're made of, how they're put together. Okay. Adaptation are just changes in that organism that allow it to do what it can do, even when you try to go after it and attack it. It'll adapt in a way that it tries to avoid getting wiped out. Okay. And attenuation is a way to render a pathogen, which is any microbe that causes disease, unable to make you sick. Okay. We use that sometimes when we prepare vaccines. Okay. Number two is talk about defenses against infection, including your immune response and prophylactic interventions. Okay, what does that mean? Well, your immune response is your body's ability to fight you getting sick, even though you live in a world that's filled with stuff that's trying to make you sick. Okay, there's things that cause disease all around us, on us, and in us all the time. But we go through most of our lives healthy. And the reason for that is because of the defenses our body's got. Okay, and there's different kinds. Some examples include our skin and their secretions, white blood cells, and a process called inflammation that we'll talk about, to just name a few. And then number three, we're going to classify microorganisms, meaning what? We're going to try and figure out what category they're in by determining what environment they like to live in, what stuff they make, and what they look like. Okay. And under the microscope, how they stain. Okay. We can stain these microorganisms so that they turn a certain color if they're in a certain category. And the reason that we do this is try and figure out how to get after them. Okay. If we can ID them, then we know how to attack them. Okay. We're going to look at microbial growth and apply the differentiation process through lab and classroom experience. Okay, what does that mean? We're going to see how primarily bacteria grow in a liquid or on a plate. Okay. And the differentiation process is essentially a set of tests you're going to do in order to find out what that bacteria is. And the way that we do that is we try to grow it on different kinds of food and see what grows and what doesn't. And we can also put stuff in the food that turns them colors when they use the food. Okay. And that helps us to figure out what they are. So it's all sort of like playing detective. Okay. This is one of the things that they will do for you if you come into the hospital with an infection, is that they will take a little bit of material from the infection, and then they're going to try and figure out with a series of lab tests what you're infected with. And the reason we want to do that is so that they can give you the right medicine to wipe it out. Okay. We're going to understand how medications affect the structure of microbes and the infection process, meaning what? We're going to figure out how these drugs interfere with these pathogens' ability to make you sick, right? How is it that they do their job? We're going to define the difference between normal flora and pathogenic organisms, meaning what? Normal flora are just the bacteria and the fungi and the protists that are on you and around you and in you normally when you're not sick. Okay. And the pathogens are the ones that can make you sick. All right. So there's going to be differences in those two in terms of what they look like, what they make, what they hook onto, okay, how they grow. And we're going to be able to determine the difference between them. And then number 7, apply standard transmission universal precautions to the chain of infection. If you've ever been in a hospital, which is full of sick people, one of the things that you run into is that people that come in maybe for a surgical procedure or something else can end up getting an infection because of the other infected people that are in the hospital. And there are practices that the hospital uses to try and cut down on that. Okay. One of the things that everybody's doing right now to cut down on COVID is wear a mask and get vaccinated. Right. So we're all aware of that because of the pandemic. And then we're going to look at the OSHA rule for occupational exposure to bloodborne pathogens. One of the things that you never want to do is accidentally get somebody's infected blood on you or in you, because then you can pick up what they have. All right. And so there are ways to avoid that, that you put into practice, whether you're part of the clinical staff or part of the general staff. Okay. The grade scale is here. All right. So you want to get at least 70% to be passing. All right. And then the breakdown is here. Right. So the tests are on Connect, the midterm and the final are going to be on Connect. Your project is going to be a disease presentation that I'm going to give you guys as soon as possible so that you can get to work on it. And what it is I'm going to give you an example of somebody that did something, like got a cut or a scrape, and then they started to show certain symptoms, right? And I'll give you a picture of the organism, okay? And then I'll tell you what other symptoms they might have. And what you're going to do as a detective is try to figure out what they have and how it should be treated, okay? And that'll be, and what will happen is you guys will take over the Zoom at that point, and I'll let you share the screen with the class and show them what you know. So you'll do like a little PowerPoint presentation on what you got, okay? And in addition to that, we're going to have something called Disease of the Week, where I'm going to give each of you a disease to look at. And what I want to know from you guys is the name of the disease, what happens to you when you get the disease, what kind of sick do you get, all right? What are your symptoms, all right? What's the organism that causes the disease, okay? Is it a bacteria? What kind of bacteria is it? Is it a parasite, etc.? Okay. And then I want to know what the treatment is and how the treatment's going to work. Okay. Then you got lab reports, and I'll send you those and you'll fill them out and send them back to me. There'll be virtual lab reports based on stuff that'll be talked about. And then I'll explain to you how I want you to approach it. There'll be a lab midterm and a final. These are what we call practical exams, okay? And I will put those together for you, and they will probably be on Connect. If not on Connect, I will send them to you, okay? And you'll complete them. The reason for this is that for the lab midterm and final, you're going to have to see images, okay? And I don't know in Connect if I can make a custom exam. Maybe I can, but if not, I will send it to you as probably a PDF file, and you'll complete it and send it back to me, and then I'll grade it, okay? And then that ends up at 100%, okay? Last day of class is 5/19, okay? And I just wanted to go through what we're doing and when we're doing it, okay? So everything that's something that's graded is in red, all right? So each week when we talk, after we talk, there's going to be an online exam that'll be open up until the next time we meet. And what you want to do is get that done and connect, and the grades will show and connect, and then I'm going to enter them in the grade book on Compass. And what you know Compass better as the student portal. All right. And I'll show you what the login is for that. And that's where a lot of the material that we're going to use in the class is also going to be posted. Okay. And it'll be available on a calendar. All right. So we're going to start with an introduction to microbes and what they're made of. And then we're going to talk about methods for studying microorganisms. How do we do that? Okay. Your lab manual is techniques in microbiology. Okay. And your textbook up here. Up here is right here. And it's Cowan. All right. And it's the 3rd edition. And in our Connect section, there will also be an ebook of that. All right. In addition to the hard copy that you've already got, unless you guys have an electronic copy already. Do you guys have the hard copy of the book or the electronic copy? Do you know?
We have the hard copy.
You got the hard copy. So with the hard copy, there should be an access code to get on to Connect. And I've sent you guys registration instructions to get into our section. And you'll use that code as a way to get in. If it doesn't work, let me know so I can talk to the rep and get you guys set up. If the code that's in your book isn't working, there is a 2-week free option until I can get the rep to get you into the Connect section permanently. Okay. All right. So we'll start out with chapters 1 and 2 in Cowan, right? And then next week, we're going to talk about some of the different kinds of organisms. Okay. We'll talk about what bacteria are, what they're made out of. We'll talk about what the archaea are, what they're made out of. And then we're going to talk about microbes that are eukaryotic, that have a structure that's more like the cells that we're built out of. Okay. And then in lab, we're going to do Gram staining. Okay. And then we're going to do Kirby Bauer. All right. And we'll talk about that. And I'll have an activity for you to go and carry out. Okay. And then there's going to be a timed exam on 3 and 4. It'll be due before we meet next time we come together. Now, I should tell you that all the Connect assignments are, they're timed in the sense that when you open them up, there's a clock that starts ticking. You have a certain amount of time to get it done. And after that time expires, that will automatically submit to me. Okay. So what you want to do with these is you want to study up before you take them, which means look at your notes, look at the podcast. This lecture is being recorded, and I'm going to send you the link to the podcast when it's done. And you can use that to fill out your notes. Okay. And I'm also going to provide you with a PDF of the notes that we're going to use so that they're easy to write on. Okay. You can print them out and write on them, or you can write on them in your computer. Okay. And then moving into the next week, week 3, we talk about viruses.
How many questions are our tests?
Come again?
How many questions are on our tests?
I'd have to look at that. The weekly exams, I want to say are 20 questions, but don't hold me to that because I need to take a peek at that to let you know. Okay. But you'll have more than enough time to finish. I've made it so that you have almost twice the amount of time to finish as you'd normally take. Okay. And then we're also going to talk about what microbes like to eat and how they grow. Okay. Then the following week, oh, and in lab, we're going to do Kirby Bauer. Okay. I'll explain to you what we're doing and how we're doing that. Okay. And then lesson 4, all right, chapter 11, which are how microbes and humans deal with each other, how they interact. And then chapter 15, which is how to figure out what kind of an infection you have. All right. Your lab activity is going to be a water pollution lab. All right. We will, if we were in an actual lab, you would bring in a water sample and we would grow it up and see what's in there. All right. But in our case, we're going to simulate that. Okay. And just as usual, there's going to be a timed online exam over 11 and 15. Okay. And then next, lesson 5, infectious diseases of the skin and the eyes and the nervous system. Okay. And then you're going to have your first disease of the week presentation, which you're going to be given the prior week. Okay. So in week 4, you're going to get your disease for you to talk about in week 5. Okay. And then there's going to be a timed online exam over 16 and 17. And there will also be a midterm exam. Okay. At that point. And that's going to go through everything up through week 4. Okay. Then in week 6, another disease of the week presentation. There will be a lab midterm that week. All right. And then in lecture, we're going to talk about diseases of the cardiovascular and lymphatic system, and then diseases that hit the respiratory system. Then you'll have a timed exam over 18 and 19. Okay. In week 7, in lecture, infections about that hit the GI. All right. And those that hit the genitourinary, be another disease of the week presentation. And then you're going to have a timed online exam over 20 and 21. Okay. Lab activities, using antibiotics to test 2 organisms and identify which ones are effective against which group. Okay. And again, that'll be a simulated activity. All right. And then in week 8, we're going to talk about how to control microbial growth. All right. What do we do in the office or in the lab or in the clinic to keep microbes at a minimum, right? Especially those that cause disease. Lab activity, we're going to look at the antiseptic and disinfectant effectiveness of controlling growth. Okay, so you're going to have some organisms that'll grow up, and then you're going to hit them with these different antiseptics and disinfectants and see how good they are at knocking it down. Okay. And then you're going to have a timed online exam over 9 and 10. All right. Then in week 9, we're going to talk about immunity, how it is that the body keeps you from getting sick. Okay. In lab, we're going to review for the lab final. Okay. And then you're going to have a timed online exam over 12 and 13. Okay. All right. And then finally, last session, okay, you're going to have, that shouldn't say lab midterm, that should say lab final. And you're going to have a lecture final that's going to be on Connect. Okay. And what we'll do here in week 10 is we're going to talk about immunity. All right. And then we're also going to have a review to get you ready for the actual exam. All right. And the other thing that I'll do with the Friday office hours is I will also provide review sessions for the weekly exams and also for the midterms and the finals. Okay. So these will be example questions. What the answers are, we'll go through it after you try and guess what the answer is. And then we'll discuss why that's the answer. Okay. So it'll be like a Q&A, sort of like a drill. Okay. Y'all know that if you want to get good at something, you practice it, right? So this is one of the ways that we're going to get you ready for the test is you're going to practice taking the test. Okay. So we're going to have a training test that's going to draw from the same kind of questions that the real test is, right? And then we're going to go through it. We're going to review it. And then that'll help you get ready for the real exam. Okay. So practice it, study it, practice it, and then do it. Okay. And that way, I think you guys will do much better on the material that we're going to talk about. Okay. And then if you want extra practice, let me know. All right. And I will do that for you. Okay. There's a place for your signature. Okay. And you can take that page and you can sign it and take a picture of it, send it back to me so that I know you've read the syllabus and understand it. Okay. So that's like the contract between you and I, right? That explains what I expect of you and what you expect of me. Okay. Important point is that you want to do these things by the due dates that are posted. All right. And I'll show you in the Connect section where those are. Okay. If you post after the due date, then there's going to be a 10% late penalty, and that's going to knock your grade down. All right. So you want to make sure and get your stuff done when you have to. Okay. So with that, let's just take a quick look at our Connect section. I just want to show it to you. So we go here. All right. And we're going to go into our section here. And then I'm going to share this with you. So we want that way. Yeah. Share that. Okay. So sign in. Come on. Wake up. There we go. Now we're awake. Use your ATA to sign in. And click in. Come on, go. Go slow today. Oh. Where's the ATA? There we go. So there's our section. Okay. And you'll see up here in this category, a midterm and a final. I haven't written the final yet, but you've got Learn Smart activities that are here. Okay. And there are, come on. There you go. Get down and stay down. There we go. And you can see here that they've got due dates on them, right? All the Learn Smart due dates are set to be due before the end of the term, right? But that's not the way I would approach them. I would do the Learn Smarts as we cover the material in class, okay? So these are just basically homework assignments that are going to help you do better on the exams, the midterm, and the final, okay? But those are open from now until the end of the term, all right? But you want to do those. Don't cram those in and try and do them all the night before the exam, okay? Because you won't be able to finish them and you'll miss out on those points. The weekly exams are here, okay? So you'll see there's a total of 9 of them, okay? And again, you will see the due dates, all right, are going to be before the next time that we meet, okay? So you're going to have 1 and 2 due before the next time we meet next week. You're going to have 3 and 4 due before the next time we meet the following week. And so they just go by, the weekly exams go by the chapters that we cover, right? 1 and 2, 3 and 4. There's usually 2 chapters per exam, okay? So you'll want to get those done after you've studied your notes and after you've taken the practice exam, okay, which I will send to you, or I will actually probably post them here, okay, as training exercises. All right. The ebook is over here. All right. So you can click on that and it will take you into an electronic version of the book where you can read and you can take notes and all of that. Okay. The browser, I would recommend using either Firefox or Chrome. Okay. Because the site is set up to work best with those. And when it pops up, what you'll see is a table of contents and the different chapters. Again, taking a while to load here. Da, 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 da. Keep going, keep going. Yeah. I understand. Fine. Open the book. Show me. Yellow highlights help you focus. Okay. Green highlights what you've mastered. Okay. All right. You can find the table of contents by typing in where the menu bar is. Progress is saved as you complete assignments. Okay, they draw on the book. Okay. All right, but anyway, here's the book. Okay. Choose your name. Good, nice. So, oh, okay. It's loading. It's meant to load. Okay. All right. That's not displaying quite right. Okay. There we go. There we go. So what you'll see here is that the Learn Smarts are set up and they draw on the textbook, okay? And it'll take you to those areas that are highlighted, right, in the textbook to give you hints on where the right answer is, okay? So I want to go to now the table of contents. Library, notes, underline. Da, 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 da. Let's see what we got here. Click on this. For me. Click on it, downloads. Yeah, it's a little slow. I know. Okay. And so what it's going to show you is an electronic version of the book. Okay. The pages will pop up and you're able to read it and highlight it and also put your own personal notes in the electronic version of the book. Okay. So that's reasonably handy. You can also study on your own. You click the other tab, the whole book is available to you, and that will allow you to study at your own pace. But it's just kind of loading real slow here. So I just wanted to show you what our section looked like and how to get at it. Okay. All right, so that's the smart book. Okay, so let's bump out of here. There, that finally popped up. And then let's talk about what we're here to talk about, which is chapters 1 and 2. All right. So we're gonna bump in and start by starting. All right. Share. And yeah. There we are. Okay. So what's the idea here behind chapter 1? Okay. The learning outcomes are listed. I'm not going to go through and read each one because we'll hit each one as we move through it. Okay. Let's start, right? Microbiology is a branch of biology, which is the study of life that happened when the microscope was invented. The microscope was invented a long time ago at the end of the 17th century by a gentleman named Leeuwenhoek. And he was a lens grinder, and he made this instrument that hadn't existed before. And he took a look through his instrument, which is a series of magnifying lenses, at a little piece of cork that he had sliced up and placed on a flat surface. And what he noticed when he examined it is these little air pockets in the cork. And he called those little air pockets cells. And that was one of the first pieces of evidence that living things are made up of cells. He looked at other living things. He looked at plant material. He looked at animal material. He looked at scrapings of his own teeth. He studied all kinds of stuff that he could look at under his new instrument, and everything he looked at was made up of cells. And so that was part of what eventually became the cell theory, which is that all living things are made of cells, and all cells come from pre-existing cells through a process called cell division. Okay. And that was a big deal, right? Microbes include bacteria, archaea, protozoa, fungi, helminths, viruses, and algae. Okay. Of the ones listed, only the viruses aren't considered to be living things, because in order for a virus to reproduce, it has to have a host. So it has to be inside another organism in order for it to make more copies of itself. So a virus is considered either active or inactive, but it's not considered to be living. Okay. Things that are living are able to grow, they're able to reproduce, they're able to move, they're able to take in nutrients and spit out waste. Okay. They have definite barriers. and they have an ability to respond to their environment in a way that makes it more likely that they're going to live. Okay. Those are basic characteristics of life, and viruses flunk the reproduction test. Okay. They don't reproduce on their own. Okay. Now there are other, you might consider them microbes that are even simpler than viruses. There are little molecules called prions that make the disease, as mad cow. Okay. And a prion is nothing but, they call it an infectious particle. And it's just a little tiny protein that you can take in by eating infected meat. And it's so small that it can move through the lining of your digestive system and into your blood and into your brain. And it'll turn your brain into jelly in about 2 days and you'll die. Okay. So it's a pretty nasty little sucker. Okay. How do you get mad cow? You eat meat that's infected with mad cow. And it turns out that most cases of mad cow come from meat that was brought into the United States from outside the United States. Okay. We feed all of our beef nothing but grain or grass, and that's federal regulation. So that's why we don't spread mad cow. Mad cow actually turns out to be spread by farmers who put bone meal in the cow feed, and the bone meal contains nerve tissue, and in the nerve tissue are these little infectious particles. Okay. The reason that they sometimes put protein in their feed in places other than the US is because the extra protein makes the cow grow bigger faster, and so you can get more meat to market. But then there's the risk of mad cow. Okay. So microbes are very easy, but also kind of tough to study, right? They reproduce real quick. Okay. Average for a bacteria to reproduce is once every 20 minutes. Okay. Now think about that compared to, say, how often a human reproduces, maybe once in a lifetime, right? So once every 20 minutes. So that's a lot of growth real quick. And that's because microbes are small and simple compared to an organism like a human that's made up of many cells and tissues, right? So it's easier to make more of the microbes more quickly. Okay. It can be grown in large populations in the laboratory most of the time. Okay. There are some microbes that can't, and they're called fastidious. Okay. They can't be seen directly, meaning what? We can't with our eyes see them. All right. We have to use a microscope, and we have to use frequently a stain, meaning a dye that turns them a color so that we can actually make them out. Okay. We analyze them through indirect means, meaning what? Well, if we can't see the microbe with our eyes, we can look for evidence of the microbe by their byproducts, right? Do they produce a gas? Do they produce an acid? Do they turn a certain chemical agent a particular color, right? And then we know that they're there. And we can see them through the microscope, right? It's kind of funny that before the microscope, we didn't understand a whole lot about why we got sick and how we worked. Okay. In the Middle Ages, for example, the barber and the doctor and the surgeon and the dentist were all the same guy. So if you were sick, you would go to him. And back in the Middle Ages, they thought that disease was the result of the balance of your body's humors. And the humors were bile, phlegm, blood, and sweat. Okay. And the fix for every disease was to bleed you. So you would go into this guy and he would say, well, you look sick, so let me cut you and bleed you. And after a certain amount of blood's been let out, I'll pronounce you cured. Right. And then he would dry his bloody rags on a white pole outside his door. And believe it or not, that's the origin of the barbershop pole. Okay. The ancient Aztecs believed that the reason that you were sick was because there were demons living inside your brain. And so what they would do was take a hammer and a chisel to the skull and knock a hole in your skull to let the demon out. And you would think that, well, everyone must have died after that procedure, but turns out no, because they've dug up graves in those areas where the skull wounds have healed over. So some of them actually survived, right? And then we used to think that people got sick because they were cursed, okay? And so we would sometimes burn people at the stake because we thought they were cursed with disease. They'd done something bad. It wasn't until the microscope, where we were able to see something we couldn't see before, that we began to understand that disease could be spread by these little things, okay? We're going to learn later about something called Koch's postulates, which are very straightforward. And essentially what Koch's postulates say is that if you see something that got infected, something sick, right? You can take material from the sick thing. You can look at it under the microscope and identify the microbes that are in there, okay? You can grow those microbes up on your own. You can infect healthy tissue. And if you recreate the disease and then re-isolate that same microbe from the freshly infected material, you know, that's probably what's making you sick, all right? And before the microscope, there was no way to do that, right? Because we couldn't see what we were dealing with. Microbes have been around for billions of years. In fact, the first life on the planet was probably a microbe, okay? Single-celled organisms that preceded the kind of cells that we're made out of came up about 3.5 billion years ago, okay? Now, I don't know if you've ever looked at the number 1 billion, but I'll just clue you in. It's a honking big number, okay? So when you're talking about change over time for life on the planet, you can almost think in terms of having an infinite amount of time to do something, okay? And you can imagine if you had all the time in the world to do something, you could probably do anything, right? If I had all the time in the world to do something, I could probably stack decks of cards from here to the moon, okay? If I had infinite time. And that's what we're talking about when we're talking about how life appeared on the planet and changed as the planet changed, okay? And that's a process that's called evolution and natural selection. And the way it works is that there are populations of organisms that have differences in their appearance, in their characteristics, and in their behavior that are the result of the information in that organism called genes, which are in a molecule called DNA, right? And when the environment changes, some of those organisms are going to be less likely to live long enough to breed, and other organisms are going to be more likely to live long enough to breed. The ones that live long enough to breed are going to pass their traits, their genes, on to their offspring, and they'll be more able to live in that new environment. The organisms that can't do that have a few choices, right? They could get wiped out, or they could leave that environment, okay? Or they could adapt in some other way, all right? But over time, what happens is as changes keep happening in the environment and we keep selecting for different traits that make it easier for that organism to deal with that environment, you change from one kind of organism into another. You become what's called a new species, okay? And the definition of a species is just organisms that can breed and make another organism that can also breed, okay? So that's why on this planet, you have all the different kinds of organisms you've got that do all kinds of different jobs in the environment, right? You've got birds that fly through the sky and feed on things like insects or small animals on the ground by pouncing on them from the air, right? You've got organisms that live in the dirt and basically crawl through the dirt and aerate the dirt and live off the stuff that's in the dirt, like maybe other insects or maybe the dirt itself. Talking things like worms and moles and stuff like that. Then you've got organisms that are good in the water, like fish and whales and octopus. Okay. And what has happened to create all those different organisms is just evolution and natural selection. Over time, through trial and error, right? You get a collection of genes together that make an organism that's real good at a particular job in the environment, which we call a niche. Okay. And that's what that organism is going to do for the rest of its time on the planet. Right. So there are three types of what we call domains that came from a single common ancestor. Okay. The eukaryotes, the bacteria, and the archaea all came from some original cell that was here on this planet. All right. And we don't know how that cell happened. Okay. We're just, I mean, a couple of guesses are it may have drifted here from outer space, or it may have just been created as a result of processes that were going on the planet at the time, which is a very different planet than we live on today. Okay. But the honest answer is we don't know because we weren't there. Okay. So science kind of has a sort of a black box at that end of life because we know what happened since, but we don't know what happened before. Okay. And we've never really figured it out. The eukaryotes get that name because they have what's called a true nucleus. Okay. U means true or proper. Let me get the pen going here. Okay. So U means true or proper, true. And karyo means nucleus. Okay. Karyo. You guys are going to take or have already taken something called medical terminology, and MedTerm teaches you about the fact that a lot of the language that we use in medicine comes from Greek and Latin. And that's something that we're going to see over and over again in this course too. And when we hit a word that is something unusual to you, we're going to break it apart and pull out the meaning. Okay. The bacteria are single-celled organisms. Okay. And they don't have what they call a true nucleus. By mean no true nucleus, we mean no nuclear membrane. Okay. And so, stop it. Sorry about that. And so these are called prokaryotic. Pro means before. Okay. Prokaryote. Pro means before the true nucleus. Okay. And then archaea, which are also prokaryotic. Okay. So these two are prokaryotic, but are distinct from bacteria. The reason being where they like to live and what their DNA tells us about them. Okay. Every living thing has DNA as part of its structure. Okay. The DNA is made up of information packets that are called genes. Which produce characteristics. characteristics. Okay. So when we look at the DNA of an archaea and we compare it to the DNA of a bacteria, they're very different from each other. The information in them is very different. And so the characters are very different. Archaea dig extreme environments like high temperature, high temp, or real acidic environments, which is called low pH. Stop that. Low pH. Okay. Or high pressure. Or high salt. Okay. So we have a nickname for them. We call them extremophiles. Meaning what? They live in places that other stuff has no chance. Okay. Phile just means loving. And so they love extreme conditions. Okay. You'll find archaea in an environment that's basically boiling battery acid, and they dig that. They're cool with that. No problem. You'll find them living in temperatures well below freezing. Okay. You'll find them living on salt deposits, where there's nothing else, just the archaea. So these guys are probably the first types of life on the planet, right? When that first cell happened, the archaea were probably the first thing to spring up. And then the bacteria followed and the eukaryotes followed after that. Okay. Bacteria and archaea are basically single-celled, while eukaryotes can either be single-celled organisms or multicellular organisms. Okay. Humans are multicellular. Humans are eukaryotes, okay, made up of many cells. Why am I doing this? Stop. Stop, stop, stop. Hash going on. Humans. Okay, multicellular eukaryotes, multicellular eukaryotes. Okay. An amoeba is a unicellular eukaryote. Okay. A eukaryotic cell is bigger and has chambers inside it that are called organelles, which are like little rooms where chemical reactions that help the cell live happen. Okay. And those little rooms keep those chemical reactions in a tiny area so that they're more effective at doing what they need to do. And they also separate chemical reactions that interfere with each other. All right. So you got a more complex setup here in the eukaryote. All right. But in terms of the number of organisms, eukaryotes are a small minority on the planet compared to bacteria and archaea. There's billions and billions of bacterial and archaeal cells all over the environment. Microbes are ubiquitous, meaning you find them basically anywhere. Okay. And like we said, wherever we look, we're going to find some sort of a microbe. All right. Pretty much. They're important because they're both beneficial, but they're also detrimental to human life on the planet. Okay. They help out a lot of things in the environment. All right. Such as they help with things like the nitrogen cycle and the carbon cycle. Okay. Which are ways that we get elements to move through an environment filled with living things, which we call an ecosystem. All right. But they can also do great damage, right? They can cause disease. So, it's not all good and all bad. And so you're looking here at just a diagram that shows you how things probably shook out, right? Around 3.5 billion years ago, life happened. Life, right? All right. Life happened. Come on. Cooperate. Why are we drifting all over? There we go. So life happened right around there. The ancestral cells gave rise to the 3 domains. And then from the eukaryotes, we got all sorts of different organisms. They show you insects, reptiles, mammals. Among the mammals include the humans, okay? But there's many different organisms, types of organisms in each category, okay? But in terms of number of cells, it's the archaea and the bacteria that outrank the eukaryotes big time, okay? It's easier to make a simpler cell. So we talked a little bit about evolution earlier, right? A theory is something that is testable, okay? Meaning that if a theory states something, that we can set up an experiment to show that the theory is accurate, all right? The accumulation of changes that occur in organisms happens because of natural selection, right? Tiny differences in populations of organisms provide an advantage for some organisms and a disadvantage for others. And the ones that get that leg up, they're the ones that are going to live long enough to breed, and their genes are going to become the big players in the offspring organisms. Okay, and the genes that don't are going to become less common. Okay, we can document this in all corners of the planet. Okay, we can see evidence of evolution in the fossil record. We can see evidence of natural selection in real time. Okay, we can do that, for instance, with bacteria as one example. And it's testable by the scientific method, right? Science works by the scientific method, which is that you make an observation, you collect data on what you're observing from sources that are already out there, and then you form a guess as to how that thing that you're observing works. You know, what's the deal? How is it doing what it's doing? And then you make an if-then statement, a hypothesis that is testable, right? If what I think is true, then when I do this, this should happen, right? And so what I do is I perform that experiment and I see whether I support or I refute my hypothesis, okay? If my experiment refutes my hypothesis, I throw it away. It's no good. But if it supports it, then I might do further testing for further support, and then I'll communicate my findings to the people around me, right? And then they should be able to, based on what I've told them, repeat that experiment and get that same result. That's the big thing about the scientific method, is it has to be reproducible, okay? If you can't repeat what somebody claims, then you call it into question, right? Well, I did what you said, but that isn't what happened, okay? And the scientific method is something you probably do every day. You just don't think about it, right? Mechanics do this when they work on your car, right? You take your car in because it's not working right. It's making funny noises under the hood, right? So you tell the mechanic, you know, what's been happening. So what does the mechanic do? The mechanic says, okay, this is the data. This is what's already known about the problem. You got noises under the hood, okay? So I'm going to start the car up, see what happens, okay? So I hear the noises under the hood, and then I turn it off and I make a guess as to what might be wrong, okay? And let's say that maybe there's he guesses that maybe there's something in the car's frame that's out of alignment, okay? So what does he do? Well, he tests to see if the frame is okay, all right, by comparing the frame on the car that he's got to deal with a normal frame, okay? He'll examine it, he'll perform certain stress tests, and he'll see whether or not his guess is right, right? And if his guess is right, then he's going to go work on the frame and fix your car, okay? And if his guess is wrong, then it ain't the frame, it's something else, okay? So you guys do scientific method every day. You just don't call it that, probably. You don't think about it, but it's just trial and error. It's all it is, okay? In science, a theory is something that's supported by hypotheses that have been tested through the scientific method. All right. Years and years of testing have not disproved the theory. Okay. So we haven't proved it wrong yet. And it's usually a well-studied phenomenon as opposed to a random guess, right? A guess is only part of the scientific method, right? The thing that makes a scientific method different from just a random guess is you test your guess, right? If you don't test your guess, then it doesn't mean anything, okay? You got to test your guess and show people how you tested it, and they have to be able to test that guess and get the same result, okay? All right. Microbes have a lot of roles in our planet, right? Photosynthesis is a process that allows a lot of life on this planet to exist, and it's the use of energy from light in order to use carbon dioxide and water to make sugars, okay, which form the structure of the organism. Plants, for instance, do photosynthesis, and the plants use those sugars to build the wood, and they also use those sugars to make starch and to make sugars that are in the plant that the plant actually uses for fuel, okay? And that sets into motion an ecosystem where some organisms will eat the plant, some organisms will eat the organisms that eat the plant, okay? And then eventually the plant and all those organisms will die, and then microbes will take over and reclaim a lot of that organism. Some of that they'll use to grow and reproduce, but some of it is going to end up back in the soil and in the water, all right? And will eventually re-enter the ecosystem. You know, these organisms are called decomposers, okay? So that's how a lot of ecosystems are set up. Photosynthesis can be anoxygenic, meaning it doesn't make oxygen, right? The photosynthesis in plants does. And it turns out that we need the oxygen that the plants produce in order to make enough energy for our bodies to function, right? With oxygen around, we make 20 times more energy than without it, okay? So a lot of life on this planet depends on oxygen to live, in addition to providing nutrients, right? So that's big. So plants do a big job for the environment. Anoxygenic photosynthesis happened before plants evolved and didn't produce oxygen. It's a more efficient way of extracting energy from sunlight, but oxygenic photosynthesis came out of anoxygenic photosynthesis and is responsible for most of the photosynthesis on the planet, right? And the reason the photosynthesis is important is because it begins all of those elements in the environment cycling through the ecosystem and keeping things alive, right? It's responsible for things like the nitrogen cycle and the carbon cycle and all the other elements that make up the building blocks of a living thing, being able to move through the environment from one organism to another. Microbes are the main forces that drive the structure and content of the soil, water, and atmosphere. They generate carbon dioxide, nitric oxide, and methane that are found in the atmosphere and help protect us from things like damaging radiation from the sun, okay, that kind of form a shield. Microbes are the most abundant organisms in the ocean. Viruses are the most abundant inhabitants of the ocean, but they're not organisms because they're not living, okay? And bacteria and fungi live often in close association with plants and help them get their nutrients and also fight disease. So one of the things, for instance, that the bacteria will do is the bacteria will take the nitrogen that's in our atmosphere, that's 70% of the air that we breathe, and turn it into a form that dissolves in water so that the plants can take up the nitrogen through their roots and the plant can grow, okay? Nitrogen, for instance, is one of the things you find in fertilizer. So you have fertile soil, the plant can grow. And the nitrogen is important because you need the nitrogen to build stuff like DNA and protein, okay? And without that, there's no life, right? It's kind of like a brick house, right? if you don't have any cement, you're not going to be able to make the brick house because you don't have anything to make the bricks cling together to build the wall, right? Without the nitrogen, you can't make the proteins or the DNA or the RNA or a lot of the other things that life depends on. So you're missing a critical ingredient. So the microbes help that get cycled through the ecosystem, okay? So how do we use microbes? Well, examples include bread and alcohol production through the use of something called yeast, which is a fungus actually. Cheese production through the use of bacteria. Treatments of wounds and lesions as a result of extracts from things like fungi and other microorganisms. We use them to mine precious metals from ores that have oxides in them, right? So for instance, bacteria, there are certain bacteria that can take rust and turn that back into iron, which is pretty cool, okay? They can clean up human created contamination, okay? In fact, I don't know if you've ever looked on the west side of town, downtown at night, you can sometimes see a smokestack sticking up that has fire shooting out of it, okay? And that's the sewer district. And the way that you treat sewage, okay, is that the sewage comes in and it goes through a strainer so that large stuff gets strained out. And then that water goes into a tank, and that the solid material that's in the water just settles. So it sits in the tank and the solid stuff settles. And then you drain the water off of that tank into another tank, and you take that and you kind of stir it up so that the air mixes with the water, and the bacteria that are normally in that water help to break down stuff that's toxic, okay? And by doing that, methane comes off, which burns. And that's what you see burning in that little smokestack, is that methane coming off, okay? And that's actually can be used as a fuel. And then you take that treated material, you put it in another settling tank, and then that water goes back into the Ohio River, all right? And then on the east side of town, I don't know if you've ever seen it, is the plant that makes our drinking water, okay? And what they have to do is they have to take the water from the Ohio River and turn it into something you can drink without getting sick. And that's a pretty amazing process too. If you ever get a chance to go there and see how they do what they do, it's really interesting. It involves using filters, using ozone, using electrical currents, okay? And they remove most of the microbes that are in the water, and then they add stuff like chlorine and fluorine, and then you have tap water that you can drink. It's perfectly safe, which is pretty amazing, okay? Considering, I don't know if you've ever seen the Ohio River, but Ohio River water is nasty, okay? Genetic engineering is another use of microbes, right? We can take organisms like bacteria and we can put genes into them using special tools called enzymes, right? To cut and paste segments of DNA together and then make the microbe take that DNA up and then use it to make those gene products. And then we can isolate those gene products and use them for things like drugs or food additives or any number of other things, okay? And that's how we do it, through recombinant DNA technology, right? Literally cutting and pasting pieces of DNA together to make a molecule that you want, and then throwing that into the microbe, and then the microbe uses the information to make the molecule that you need, okay? Bioremediation uses microbes that are already present or introduced intentionally to restore stability or to clean up toxic pollutants, okay? The vast majority of microbes are harmless to us, but pathogens make disease. There's about 2,000 different microbes that can do that, causing about 10 billion infections a year all over the world, okay? Examples of re-emerging diseases include AIDS, Hep C, West Nile, tuberculosis, and then we've got this thing that was created in the wild that's hitting us right now, which is COVID, which is a cold virus that has been assembled from different sources, either deliberately or naturally. Okay, we don't really quite know yet how COVID happened, but it's here now, and it's a unique pandemic. Okay, associations between non-infectious diseases and microbes include gastric ulcers that are caused by something called Helicobacter. Helicobacter is everywhere. You eat it, you drink it, you breathe it in, right? But in some people, because of their genes, they have a surface on their stomach that these helicobacters can cling onto. And when they do, they irritate the stomach lining and they cause inflammation. And that, along with the action of stomach acid, carves a hole in the stomach and starts to bleed, and that's an ulcer. And if that hole gets all the way to the other side, that's a perforated ulcer. And then your stomach acid can leak into your abdominal pelvic cavity and start to tear up your own organs. And that's an immediate medical emergency. So because we now know that the Helicobacter is the bad guy here, we can treat a lot of ulcers through antibiotics. Okay. Cancer can be associated with bacteria and viruses. Cancer is just a normal cell that has undergone a change in its DNA that makes it a cell that just divides on its own without helping the body. And what that cell mass does is it steals oxygen and nutrients from the blood at the expense of everything in our body that's healthy. And that ends up basically starving us of what we need to live, and we die. Okay. Diabetes is associated with the Coxsackie virus. Okay. Schizophrenia associated with the Borna agent. All right. An increasing number of patients have weakened defenses that can be infected by microbes that normally don't make us sick. Okay. An example is somebody on chemo. Okay. When you're on chemotherapy, you're taking drugs that hit your tissues so they don't divide as rapidly. And the idea is that because the cancer divides rapidly, the drug will hit the cancer and the cancer will go away before the patient goes away. And sometimes it works and sometimes it doesn't. But when you're on chemo, your immune system is also hurting. And the result of that is that you end up getting infected by stuff that normally wouldn't make you sick. Okay. And so you get stuff like thrush, which is fungus that can grow in the oral cavity, makes a white fuzzy tongue. Okay. And is very causes discomfort and pain. Okay. That's just one example. We're seeing an increase in microbes that are resistant to drugs, and there's a reason for that. Okay. And it's usually the improper use of drugs when you get them from your doctor. Okay. So you can see some of the top causes of death over all diseases, both worldwide and in the US, right? And you'll note that a lot of them are the result of microbial infection. All right. So understanding them and knowing how to fight them is going to cut down on a lot of death. Okay. Now we talked about eukaryotes and prokaryotes, right? Eukaryotic cells are complex and contain organelles and other structures, and they're bigger than like a bacterial cell. Okay. Some eukaryotes are microbes and some are visible to the naked eye, such as multicellular eukaryotes. Examples would be a dog or a cat or a human or a cow or a tree. Okay. Those are all multicellular eukaryotes. Prokaryotes, like bacteria and archaea, are much tinier. Okay. You have to have a microscope to see them, and even then they're kind of tough to see. They don't have organelles, and they're all microscopic. Okay. Viruses are not living entities. Okay. They are infectious particles that contain genetic material, and they can't be seen with a light microscope. The only way you can actually see a virus is to use a special device called an electron microscope, which is a huge instrument that costs millions of dollars, and you generally just get one of those per institution, and everybody shares it. And it uses a beam of electrons in order to see something so tiny. And viruses are actually, they're kind of pretty to look at. They look like Christmas ornaments, but they are essentially genetic material with a protein box around them, right? And then there will be in some of them little spikes or protrusions that are part of the way they hook up to their host cell and invade it, okay? They usually attach outside the host. They introduce their genetic material, and that takes over the host cell and turns it into a virus factory. And so that does two things to that host cell. It makes it incapable of doing its job, but it also makes it spread the virus, right? And so that's how viruses are able to rapidly reproduce and cause disease in an infected individual. Okay, so next section, we're going to talk about historical aspects of microbes, right? Back before we understood about the cell theory, we used to believe in something called spontaneous generation, which stated that invisible life forces present in matter led to the creation of life, right? Even after the discovery of microbes, abiogenesis was still something that science believed, right? Well, we have to kind of give a nod to spontaneous generation in this sense, right? Whatever that first cell was, wherever it came from, that was spontaneous generation. That was life from non-life, okay? But everything since then comes from cellular reproduction, right? Biogenesis is the understanding that living things only come from other living things, okay? So now we get into Louis Pasteur, okay? Pasteur was a clever guy, all right? And he was one of the people who believed in biogenesis. And he studied the role of microbes in the fermentation of beer and wine. And he used these special flasks in order to demonstrate biogenesis, right? He would fill the flasks with sterile broth. Think of it like the liquid part of chicken soup, okay? And he would put them into sterile, long swan neck tubes, right? And he would sterilize them using heat, right? So that nothing could grow. The flasks were exposed to dust from the air and showed microbial growth, all right? Whereas those that weren't didn't, right? And so the experiment is here. And what you can see, right, is that as long as the broth stayed in the flask, there was no way for the dust and what he thought were the microbes to get up there and get in to the broth, right? So that's a trip that the air is not going to make, okay? Stop it, funky. How are you doing this? Here we go. All right. All right, it's probably going to end up stopping right around here, okay? But then he broke the neck off of one of the flasks, and so the dust could drift in, and it began to grow in the broth, and the broth got cloudy, right? Whereas if he kept the neck in place, the broth remained clear, nothing grew, right? So that was proof for biogenesis, life from life. Stop it. Genesis. There we go. Biogenesis. Robert Hooke studied household objects, plants, and trees and described cellular structure. Okay. And Leeuwenhoek was our microscope guy, right? Manufactured the simple microscopes. We started the revolution, observed what he called animals in a drop of water. He didn't know what they were yet. And observed animalcules that he scraped from his teeth. Okay. He made about 250 microscopes that would magnify to 300x. So the image you would see is 300 times bigger than the actual object. And so you can understand why we didn't believe in microbes until the microscope came around, right? Humans believe what they see. Okay. If I can't see it, it isn't real, right? And now suddenly we see it. And so now we know that it's real and we know that it can affect us, right? Cohn discovered and described heat-resistant endospores. What's an endospore? Well, it's sort of like the bacterial equivalent of a seed, right? So an endospore is this little particle that kind of waits around for a good environment to crop up so that it can turn around and sort of hatch and generate new microbes that can reproduce and grow and move and all that stuff. Okay. And endospores are tough little suckers. They can withstand extreme conditions without getting injured. So they're kind of interesting in that way. So like a seed can do that, right? A seed that's been found in the Egyptian pyramids has been planted in fertile soil and has grown, right? That's millions of years. So something in that seed was still alive, right? And that seed grew when the proper conditions came around. Semmelweis described the importance of hand washing and preventing disease in a hospital setting. And Lister used antiseptic techniques in surgery. Now, I know that this is going to kind of blow your mind, but there was a time when if you needed surgery, you'd come in, with an infection or an injury, and the doctor, without washing his hands, would cut you open, do his thing, sew you back up or whatever, and then say, okay, good luck. And most of those patients ended up dying of massive infection, right? Because they didn't understand about germs and disease yet. Okay. And with the advances of these two gentlemen, we now understand that it's important to wash your hands and sterilize your instruments before you go in there and start doing your work. Okay. There's something in probably every bathroom in the United States named after Joseph Lister. Does anybody know what that is? You gargle with it?
Listerine.
There you go. See that? Listerine. Exactly right. The germ theory of disease is what led to Koch's postulates, right? Pasteur invented pasteurization, conducted the first studies linking human disease to infection, right? And then postulates by Robert Koch established the ability to identify that a particular organism causes a particular disease, okay? For instance, he showed that anthrax was caused by Bacillus anthracis in 1875. And he did that partly by using the microscope and also by using the ability to grow up the pathogen in an independent medium and reinfect healthy tissue, okay? So you can see here some of the recent advances. I won't go through all of these, but they're in your book, okay? And so what we can do now is let's just take a 10-minute break and we get something to eat, something to drink, and then we'll come back and finish the back half of this chapter, okay? So I'll see you guys in 10 minutes. All right. Okay. We're back. Next set of learning outcomes. Talk a little bit about what cells are made out of. Talk about some important biological molecules. Talk about protein structure. Okay. So a lot of things that are critical for life to happen, right, are building blocks called molecules. And these molecules have carbon in them and hydrogen and often oxygen, sometimes sulfur and nitrogen, right? Those are the pieces of these big molecules that make life happen. And the four main kinds of molecules are carbohydrates, lipids, proteins, and nucleic acids. And you've probably seen these before in A&P. Okay, talked a little bit about them. Quick description, right? Carbohydrates are sugars, right? And their primary use in the body is for fuel. Okay, but they can also be used as structural pieces, right? They're part of the structure of DNA and RNA. They're part of the structure of the sugars that are on the surfaces of a lot of cells that serve as ID tags, right? And sometimes they are actually attached to other molecules like proteins in order to help them work or help them go to the place in the cell they need to go. Okay. Lipids are a group of molecules that don't easily dissolve in water. So they can be things like oils and lard and butter are examples of lipids. The main lipids that we're interested in are something called cholesterol and triglycerides. Triglycerides' main function is to store fuel in the body and to be a source material for phospholipids, which are building blocks of cell membranes. Okay. And cholesterol's main function is to control how phospholipids move around in the cell membrane. So they kind of act like a doorstop. And in the human body, they're an important part of bile salts and steroid hormones. Okay. And then proteins are just the tools of life, right? And they're made of building blocks called amino acids. And they do every job in the human body that you could think of as a life process. Okay. Nucleic acids are built of nucleotides and they are information molecules. They contain the blueprints used to make the proteins. Okay. Polymers are basically chains of molecules repeated one after the other, and they're made of monomers, right? So the monomer of a nucleic acid is a nucleotide. The monomer of a protein is an amino acid, okay? And the monomer of a complex carbohydrate would be a simple sugar, okay? So just like links in a chain link fence, okay, you make these large molecules by hooking these things together and forming chemical bonds between them, making long chains, okay? So carbohydrates, right? Combinations of carbon and water represented by, come
Are
again?
these PowerPoints, are we able to print these PowerPoints out?
Yeah, hold on just a second, and let me give them to you in chat. Hold on. So I'll get these in chat for you real quick. So there we go. And name. And there we go. And there we go. There we go. So here comes chapter 1. Okay. And you should see it in chat. And then let me get
We have to go into the chat to print them out. We
Yeah.
can't go on the portal.
They're not in the portal yet. I'll put them in the portal, but you can get them off the chat right now. And then let me go here. Da, da, da. Let me go to number 2. There we go. And there's number 2. We got it. So you should see them in chat. If you don't, I'll email them to you. Okay. All right. So all sugars end with the suffix "-ose." Examples: glucose, galactose, fructose, lactose. Okay. A hexose is a 6-carbon sugar. A pentose is a 5-carbon sugar. Glucose is probably the most important of these monosaccharides because it's the building block of starch and of cellulose and of chitin, okay, which is what the insect exoskeleton is made out of, and also the exoskeleton of crabs and crayfish and lobsters. Okay. Fructose is fruit sugar. Okay, you get that from citrus fruits, as well as things like apples and pears. Okay, and again, most of these molecules are fuel; they're for energy. Okay, all right, moving over. A polysaccharide is just a chain of monosaccharides, right? So examples, right? Agar is made up of, is a polysaccharide used in culture media, okay? We're going to do some labs where you're going to grow microorganisms on a plate of agar, right? The agar will have polysaccharide in it to provide essentially a solid surface. And then there's also going to be other stuff dissolved in the agar that the bacteria will feed off of. Okay. And then sometimes we'll put in the agar special dyes that will turn colors when certain bacteria grow on them, right? Like they might turn red or they might turn blue. Okay. Chitin is a cell wall. Find it in fungi and you find it in the shells of crabs and lobsters. Peptidoglycans are part of the bacterial cell wall. Bacteria has a cell wall that protects it from bursting if it moves into an environment where water will enter it more quickly than it leaves it. Okay, that's called a hypotonic environment. And so this keeps the bacteria from exploding. Okay. Wow. And then lipopolysaccharide is part of a gram-negative cell wall of gram-negative bacteria. Okay. And all of these have sugar pieces to them. Okay, saccharide pieces to them. The glycocalyx is important in attachment of cells to each other or as a site for receptors. Okay, receptors are proteins that are stuck in the cell membrane that attach to other molecules. And then by doing that, the receptor changes shape and it causes the cell to respond to that molecule in a particular way. Okay. Lipids, right? You can see an example here of a triglyceride, right? An important storage lipid made of 3 fatty acids and 1 molecule of glycerol. And it's stored as a long-term fuel reserve and yields 2 times as much energy per gram as other storage molecules. So this is why a lot of organisms store extra calories as fat, right? Because you get a lot of calories in a tiny volume. Is very effective at allowing you to basically keep a pantry in your body, right? Continue to make energy as you need it. Okay. For instance, in our body, right, lipids are important as insulation, as fuel storage, as padding and protection. Okay. We talked about phospholipids, right? These are the building blocks of the cell membrane. And what they do is they form something called a semi-permeable barrier around the cell. What does that mean? it's just like a fence that'll let certain things through and other things not. Okay, like in a chain link fence, you could maybe shove a stick through the holes, right? But a log won't probably get through. Well, the same thing is true with a phospholipid membrane, right? The phospholipids will form what we call a bilayer in the cell membrane. And let me draw that, what that would look like. But the heads would be here and the tails would be dangling off, right? And then their mirror image would be here and these tails would be dangling off. Okay, just like, you know, kind of a schematic of this drawing right here, right? And this thing just repeats and repeats and repeats on the surface of the cell, right? And those tails are greasy, they're hydrophobic, so they don't like letting charged or polar stuff get through, okay? Which means basically that certain molecules are kept out unless they go through a special tunnel or a gate, right? And other molecules can go right through, okay? But this part is hydrophobic, meaning water fearing. Okay. And then this part, the heads are hydrophilic, meaning they like water. Okay. So only certain stuff can get through this barrier. This would be what a cell membrane would look like. Cell membrane. Okay. Abbreviation. Right. And that's important for the cell to work. For the cell to function, it needs to be able to let in stuff that it needs and get rid of stuff it doesn't want, and not just let anything go in and go out. Okay. So that's the idea behind a phospholipid. And those are made from triglycerides, right? Steroids are a complex molecule found in cell membranes and used to make hormones. For the purposes of this class, the most important use is in the animal cell membrane and in cell membrane deficient bacteria, okay? And they serve as a way to keep the phospholipids from drifting around too much. And that lets certain proteins that are stuck in the lipid bilayer stay essentially in one place so they can work together better. Waxes are formed between an alcohol and a saturated fatty acid, and they're important in waterproofing, such as in fur, feathers, fruits, leaves, and human skin. And they're found in the cell wall of bacteria that cause tuberculosis. And because of that, they're more able to cause disease, okay? Because that wax forms a barrier that our defenses don't get through very easily, okay? Proteins are the tools of life, right? Proteins do every job you can think of that is a life process. They defend against disease. They make chemical reactions happen more quickly in the form of enzymes. And they tell the cell what's going on outside in the form of receptors. And they provide strength to the cell's overall structure as what they call cytoskeletal components, like microtubules and intermediate filaments and stuff, okay? And so that's really neat. And a protein can do all of that because it's built of building blocks called amino acids. There's 20 of them. And those amino acids can be put together in any length and any order to build any tool, which is really nice, right? And the information for doing that comes from the DNA, okay? The DNA gives that information to the RNA, and then the RNA is used by an organelle in the cell called a ribosome to build the protein using the instructions that's in the RNA. So the RNA is like the temporary copy of the blueprints. And so we put these amino acids together in the right order, and we make all these neat tools that let the cell do its work, okay? There's different levels of structure that we will get into in just a second, okay, that allow those tools to do their job. So let's take a listen to that now. Hopefully, oh, are you gonna?
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
And those levels of structure are primary, secondary, tertiary, and quaternary. Meaning what? The primary structure is just the length of the polypeptide chain and the order that they're hooked up. The secondary structure is the shape of certain parts of the chain as it begins to coil or fold. All right. And the tertiary structure is the way that those coiled or folded parts of the chain lie on top of each other. And then the quaternary structure is different chains fitting together like puzzle pieces so that you build an even more complex tool. Okay. Because the amino acids are, they fall into 3 groups. Okay. They can either be polar or charged or non-polar. All right. And we can put them together in any order and any length. We can make any tool you can imagine. So I like to think of it like Legos, right? Legos have a few basic pieces, but then depending on how you hook those pieces together, you can build anything from a cube to the Empire State Building. Okay. And that's kind of what we're doing here with the building blocks of proteins, which are the amino acids. And these are just some of the jobs that the proteins can do, right? The protein is going to be working in a particular environment, meaning it'll work at a particular temperature, a particular pH, okay? And that's going to allow it to keep the shape that lets it work. And if we change those conditions, then the protein can do something called denaturing, where the folded up chain will unfold and then the protein won't work anymore. And the result of that is that whatever that job that protein's doing is going to stop, right? It would be kind of like if I got into your toolbox and I melded your wrench, right? Well, you couldn't use your wrench anymore. It's the wrong shape, okay? That's exactly what happens when we mess around with protein structure, okay? Now, the information to do all this comes from nucleic acids, DNA and RNA. So let's learn a little bit about them.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So these are information molecules, okay? The building blocks are the nucleotides, and there's 4 of them, and they could be arranged in any order and any length, but depending on how you link them together, you generate different blueprints for building different proteins, okay? It's kind of like the alphabet, right? You take the letters of the alphabet, and depending on how you arrange them, you can make a novel or a journal article or a comic book, right? You can write anything, okay, just using the letters of the alphabet. So the same thing is true with DNA and RNA. The letters of the DNA and RNA alphabet are the nucleotides. In DNA, those are A, T, G, and C. And in RNA, they are A, U, G, and C, okay? And the way that the DNA is set up is that on one strand, right? If there's an adenine on the opposite strand, there will always be a thymine. And if on that same strand there's a guanine, on the opposite strand there will always be a cytosine. Okay, and that's called complementarity. And the idea behind complementarity is that when you make new DNA, one of the things that happens is that those two strands will unwind, they'll unzip, all right? And you will use the information in the old strand to build the new strand, right? So there will be tools, enzymes that will come along and walk down the old strand and put in the complementary base in the new strand. And when it's all done, what you end up with are 2 new DNA molecules, double-stranded, that have the same information as the original one. And that's important because that allows us to send that information to the next cell. And so it can use it to look and act like the original one, unless there is a goof. And goofs are called mutations, okay? And a mutation can have one of 3 effects, right? It could be beneficial, which means it helps the organism live long enough to breed. It can be bad, which means it makes it less likely that the organism will live long enough to breed, or it might not have any effect, okay? And mutations come from just mistakes in the enzymes that build DNA. Sometimes they mess up. It can come from being hit with radiation, right? Or being hit with chemicals that attach to DNA. They're called mutagens, right? And it can also come from viruses that like to stick their DNA into the organism DNA and change it, okay? And so those sometimes produce differences in the organism that can work with evolution to generate new characteristics, right? New organisms. So mutation is what we call the driving force. Okay, so from DNA, right, which would be like the master blueprint, one of the things we do is we make a copy of the master blueprint in order to generate the tool from those blueprints by making protein based on those instructions, right? So the gene in the DNA is going to be used to make a messenger RNA molecule that's going to contain the temporary copy of the information to build the protein. And then the ribosome, we're going to hop onto the messenger RNA molecule and build the protein 1 amino acid at a time using the information in the message. And then that protein's going to be let go, and it's eventually going to be sent to the area in the cell where it's going to do its work. Transfer RNA is a little different. Transfer RNA is a molecule that's attached to an amino acid so that the amino acid can be brought into the growing protein chain in the right spot. Okay. So it's like a little handle that's on the amino acid that the ribosome can grab. And then ribosomal RNA is sort of what holds the ribosome, which is the protein building factory, together. So it's like glue. Okay. RNA can also play a role in DNA regulation. We'll get into that later, right? Now, ATP is our energy molecule, right? This is the form of energy that's immediately available to do work for the cell. And the reason for that is that these phosphate groups down here, let me pin up. Okay. These phosphate groups here, right, have negative charges on them, but they're held together by covalent bonds, all right? And you can see those bonds here, right? There's one of them. There's another one, okay? And when you crack those bonds, energy comes flying out, right? And that's harnessed by other tools to do a job, kind of like gas in your tank, okay? So the idea behind the way that energy moves through the cell is that you got nutrients, right? Which are basically the food, what the cell is eating, nutrients, right? The cell takes those in, okay? Stop drifting around like that. The cell takes those in, and then is gonna break down the nutrients. Okay, by cracking chemical bonds that hold the nutrients together, and then it's going to send that energy to ATP. Okay. And then the ATP provides the cell with power. Okay. That's how it happens. Okay. And heat is made at each step, right? Bacteria and protozoa are single-celled organisms, while animals and plants are made of lots of cells, right? The cells can have different shapes, okay? They all contain cytoplasm, which are the guts of the cell, surrounded by a cell membrane. And then somewhere in the cell is DNA, the information molecule. There's also ribosomes to make protein, okay? They're very complex in their organization. We're going to learn more about the cells later on. Eukaryotic cells are found in animals, plants, fungi, and protozoa. And they have those organelles, some of which are surrounded by membranes and others that aren't, that do particular jobs, all right? Bacteria and archaea don't have a true nucleus because there's no nuclear membrane inside these cells. They have a complex structure and they can do almost everything that a eukaryotic cell can, but the tools are a little bit different, okay? They're proteins, but they're different proteins, okay? All right, we'll talk a little bit about classification, okay? One of the reasons that we like to classify things is to make them easier to deal with, okay? An example would be your mail, right? Your mail comes into your mailbox, right? And you could just take all that mail and throw it into one giant pile, right? And you could probably still use that pile, but it would take you more time and it wouldn't be as effective. But you could also take your mail and you could classify it, right? You could have one stack for bills, one stack for letters from friends and family, and then one stack for, junk, right? And that allows you to make a more effective use of that same mail, right? You can reach into the bill stack and whatever you pull out is something that you have to pay right away, right? So that's how you deal with that stack. You can reach into the personal correspondence stack and pull out anything, and that's something you should probably write back about because that's a friend or a family member, right? And then you know what to do with the junk stack, right? You pitch it, okay? Same thing with taxonomy. By organizing living things into categories, we can deal with them a little bit better, okay? We know certain things about them because of the category that they're in, okay? Nomenclature is just a way to name these different organisms, and classification is the arrangement of these organisms into a hierarchy, generally organized in terms of how related they are to each other, how similar they are to each other, kind of like a family tree, okay? And identification is the way that we determine where to put that organism, right? The assignment of scientific names and taxonomic categories to individual organisms is called nomenclature. And binomial nomenclature is a unique name for an organism that no other organism has, okay? And it's always written as genus and species, okay? Scientific names are italicized when they're written in print, and they're underlined when they're written by hand, like shown here, okay? When the name is abbreviated, the genus name is abbreviated to the first initial, followed by a period, and the full species name is written out, right? So Escherichia coli would be written E. coli, right? Pseudomonas aeruginosa would be written P. aeruginosa, okay? Just a couple of examples. When we do classification, we organize living things into descending ranks, with the most general being the beginning part and then the most specific being the ending part, okay? And so that's the lineup, right? Domain, kingdom, phylum, or division, class, order, family, genus, and species, right? So it goes, dumb King Philip came over for good sandwiches. Hold on just 2 seconds. Okay. So this is, again, just a way to put these organisms into different groups so that we can deal with them more easily. So some examples are shown here, right? And see how the categories go from real broad to very specific, right? Very, very specific. And the way that we determine where you go is by looking at your characteristics, right? How many limbs do you have? Are you made of one cell or many cells? Do you have scales or feathers? Okay. This would be mostly for multicellular organisms. And so we use those pieces of information to eventually put you into a category, right? A specific category. Phylogeny is just the scheme that represents the natural relatedness between groups of living things. And we've talked about evolution, right? Evolution and natural selection is just how we get to the variety of life that we see around us every day. Okay. And that's just working with those characteristics to pick out the organisms best suited to live in that environment and allow them to breed, right? And the organisms that aren't well suited either die off or they leave, right? Or they adapt in some other way. Charles Darwin and Haeckel proposed two kingdoms, plants and animals. Haeckel then later added protozoa or protista and Monera, right? Whitaker added fungi in a period from 59 to 69 and based it on structural similarities and differences, okay? And so now we have a different type of approach. We have something called the Woese-Fox system, right? And what is that all about? Well, it's different domains. And how do we determine who goes in what domain? Well, we use information in the DNA. We have the ability now to pull DNA out of anything and get its sequence, meaning we can read the novel of that organism. And then we can compare it to other novels and see how similar they are. And the more similar that information is, that's where that organism goes. That's how we determine which domain you're going to go in. How similar are you determines where you go on the tree. Okay, and so you can see examples here of the different domains, and there's branches coming off of those domains that would be kingdoms, phylums, and so on. Okay, so it's just an information-based approach, right? And the reason that we can do that today kind of easy is not only can we sequence, but we can actually look at little regions of DNA that are major indicators of what the thing is, and we can amplify them using a technique called PCR. Right now, for instance, everybody's doing the COVID test, okay? And what the COVID test is they put that nasty swab down your nose and they hit you in the back of your what Eric called nasopharynx, very back part of your nose, and then they take that little bit of mucus, and in that, if there's virus, right, what they do is they take that mucus and they amplify it using a tool called an enzyme and building blocks of DNA and an instrument called a thermocycler. And what that does is that makes multiple copies of little tiny regions of the book of life for that organism, okay? Or in the case of the virus, the book of the virus. And they see whether those pieces of information are there. And if they're there, then we know that the organism's in there. Okay, we can do the same thing with living things. We can pull the DNA out and we can amplify it, and we can compare that to other DNA and see where that organism should go and do it pretty quick. Okay, and that part of that technique was actually developed in Cincinnati at the University of Cincinnati because of the discovery of a DNA building enzyme called Taq polymerase. Okay, I actually knew the guy that discovered it and characterized it. And now it's used for things like crime scenes and forensics and all kinds of stuff. Okay, so it's a way to figure out who's who really fast. Okay, so with that, I know we only got through chapter 1. We'll break here. We'll come back for lab, and we'll hit chapter 2 in lab today, so we get through our stuff, because the lab on street plate is going to be kind of short. Okay, and that'll keep us on track. We'll have covered everything. The podcast for this will be a YouTube link that I will send you guys as soon as it processes, and then you can go back and use that and your notes to complete anything that you didn't catch the first time around. And it's actually a good idea because that helps you sort of hear the material again and keep it in your head, right? It's a good way to study. So before we break and then come back at, I think it's 1 o'clock, right? Does anybody have any questions? We good?
The email that you sent with the attached syllabus and the
Yeah.
I didn't have any attachments.
Oh, you didn't have any attachments. Okay. Well, then I will resend. Okay. Yeah, I'll resend so you get everything. Actually, the registration and the syllabus are also in the chat right now. So if you can pull them off the chat, you got them. You should see a file from me to everyone, registration details and syllabus, as well as
Don't
the
have
lecture
it.
notes. You're not seeing the file?
No.
Oh, that's weird.
I got it.
You got it out of chat? Okay.
Yes.
Okay. Yeah, are you on a computer? You look like you're on a computer, right?
Yes.
Okay, that's probably the difference. If you're on a computer, I think you probably see it easier. But you should be able to go to chat and you'll see a message from me to everyone. It's just an attachment. And it'll say chapter 2 lecture mod PDF, chapter 1 lecture mod PDF, syllabus Mike 212, and registration. Okay. But like I said, I'll send that to you too through just regular mail. Okay, because it looks like the attachment function in Compass isn't working for everybody. Okay.
Okay.
All right. Any other questions aside from that? We good?
Yeah.
Okay.
Yeah.
I'll see you guys at 1 o'clock. All right.
Thank you.
All right. You're welcome. Thank you for coming today.